Hello, my name is Wang. I'm a member of the Biomet Lab, and my project is optimizing plasmid delivery in Jurkat cells using acoustic-electric shear orbiting Poration. Cancer is a deceptive illness. This is because cancer is able to hide its antigens, the proteins that our immune system uses to identify it. So, how do we teach our immune system to fight back? This is the main premise of chimeric antigen receptor or CAR T cell therapy. This is a therapy where we isolate T cells from a cancer patient, genetically engineer them so they express chimeric antigen receptor, and then re-inject the CAR T cells back into the patient. Chimeric antigen receptor allows the T cell to bind specifically to that patient's cancer tumor cells and destroy them. The only approved FDA technology to transfect cells with the CAR transgene so far is viral vectors. However, viral vectors are expensive, time consuming, and can even cause an immune reaction. This is why the Biomet Lab is working on an alternative method to transfect cells with the CAR transgene called acoustic-electric shear orbiting Poration, or ASOP. In ASOP, instead of using viral vectors, we combine two main forces to transfect cells with any genetic cargo. First, we use shear forces generated by an acoustic field to open pores on the cell membrane. As the cell passes through a microfluidic channel, it is exposed to shear forces generated by vortices. Next, we widen the pores using an electric field. This electric field widens the pores and allows any genetic cargo that is also flowing through the microfluidic channels to flow through the membrane into the cell and transfect the cell. In my project, we hypothesized that we could optimize ASOP to deliver green fluorescent protein or GFP plasmid into Jurkat cells, which are a type of immortalized T cell line. We observed our results using flow cytometry to determine how many cells survived the ASOP procedure using a DAPI stain, and we also analyzed how many cells were successfully transvected with GFP. The cells that were successfully transfected with GFP plasmid would glow bright green. We analyzed the results and found that the average viability was over 90%, which means that most of the cells survived the ASOP procedure. Despite having high viability, though, our ASOP still needs to be optimized for transfection efficiency, as all of our trials had below 10% transfection efficiency. In the future, we would like to increase this number to over 80%, since CAR T cell therapy needs an average of 50 to 100 million CAR T cells for every patient. We will also continue to work on primary T cells instead of Jurkat, increasing throughput, and also using the CAR transgene so that we can make a CAR T cell therapy more accessible to everyone who needs it. Thank you.